So I thank Dr. Gitanjali Misra, convener of this conference, for asking me to share the session. And I request all the speakers of this session to stick with the time. And there are 4 speakers. I invite Dr. Kunte to deliver his talk. He is from NCBS, Bangalore.
Thank you, sir. Can everybody hear me all right if I step away from there? Good. All right. It's a pleasure to be here. This is my first ISEB meeting. Thank you, madam, for inviting us and giving me a special slot as an invited speaker, which allows me to speak a little bit more about my lab's work. And I'm going to be talking about a species that I've studied for the last species in that group for the last nearly 20 years through my PhD, postdoc, and in my lab. And the real heroes of today's talk, most of the work that I'm going to present, are Riddhi, who is the primary author on the paper from which I'm going to present work. She also received TIFR's Best Thesis Award for the work that I'm going to present. So she has done phenomenal work in this. Equally important in handling all the genomic work that we are presenting was Saurav. He was another PhD student in the lab. And Atulya is in the crowd somewhere today. So she did a lot of phylogenetic work, which was also pretty amazing, because without which we couldn't have dated most of the things that I'm going to talk about. And Muktai, who was an intern in the lab for like 6 or 7 years, starting from her 12th grade. So she came to lab really early and she really blossomed as an evolutionary biologist in the lab. So this is the work that they had done. This is the primary paper based on which I'm going to present the story of evolution of polymorphisms. This follows from several papers that we have published. There is one on, I do not have the pointer. This second paper looks at speciation. I think that would also be of a lot of interest to people here. Then Riddhi has one more paper more on the developmental genetics side of it. I'm not going to talk too much about developmental genetics today. We'll be talking more on the evolutionary side today, which is the primary work that she has done, and the analysis of molecular evolution of one particular gene that we are going to talk about was done largely by Saurav, and that was also pretty amazing work, starting from the first discovery of this gene, which was doing my postdoc. All right. So doing the modern synthesis, one of the primary architects of it, JBS Haldane, came up with a pretty brilliant idea. This was exactly 100 years ago. This was published in 1924. So he observed in a theoretical paper that novel adaptive mutations under strong selection in a large population will rise rapidly to a high frequency of fix. They will sweep through the population if they are genetically dominant over wild type. This is because even partially dominant mutations are exposed to selection right away. Remember that if a mutation is recessive, then it's not going to get exposed to selection until it rises to a frequency high enough to produce homozygotes, right? So a recessive mutation will bounce off like this under drift. It might get fixed eventually, or it might get lost from the population. But if you have a dominant mutation, the blue line here, from the beginning of that mutation, this is going to get picked up by selection, right? So its frequency will rise very, very rapidly. And therefore, what you would expect, this is the last point, you expect that strongly beneficial mutations that sweep through populations would often be genetically dominant. This is what Haldane had predicted at that time. This is now known as Haldane's sieve. Now, for ordinary traits, strongly beneficial dominant mutations would sweep to fixation. So if you're looking through a population, a history of a particular allele, you know what is existing there when you do a genome sequence or gene sequencing. What you do not know is what the wild type was. So when this kind of selection happens and things go forward, then you do not have any evidence of the wild type, right? So it's very, very difficult to study Halden sieve in nature. This is one of the reasons why if you search Halden sieve, there are very few empirical papers on it. Most of what has been written on Halden sieve is theoretical. But we realized in the lab that in ecological polymorphisms, and I'll explain what those are, the different mutations and resultant forms, ecological forms that you see, they confer some fitness advantage that helps them persist in the population. So if you look at the diversity of different alleles that you have, one of them might be a wild type, or at least you can have some parent-offspring relationship in those alleles. And when you have that history, you can actually see whether subsequent forms are actually sequentially dominant, as predicted by Halden. So we then asked, can we use these nature's grand experiments of the ecological polymorphisms that you see to test this idea? So what are ecological polymorphisms? These are two or more distinct forms in a population. The pin and thumb forms in the primula flowers is something that has been studied for, again, nearly 100 years, initially with very basic genetic techniques and increasingly with more genomic data sets and genetic manipulations. You have ladybird beetles, you have ruff, so from plants to insects to birds and many other organisms, you see this kind of ecological polymorphism. It's surprisingly very common. So if you look at an adaptive landscape, you can imagine that different forms of the same species are sitting on different adaptive peaks. And because they're on different adaptive peaks, they avoid competition, and all of them have a certain fitness advantage because of which they're maintained in the population. So balancing selection, negative frequency dependent selection, and heterozygote advantage, these are some of the mechanisms by which you can have polymorphisms maintained. And these stable polymorphisms can be strongly selected, but they may not cause speciation, right? Because of the way the genetics works as well as the selection works in these cases. However, polymorphisms are a transient phase during a sympatric speciation event. You can imagine how that works out. So polymorphisms can lead to speciation under some conditions, but not in case of stable polymorphisms. All right, I'm known as a butterfly guy, so of course I think more about butterflies and how we can use that system to ask fundamental questions in evolutionary biology and ecology. So this is one of my earliest papers from my PhD, during which I wrote about evolutionary mechanisms that might maintain mimetic polymorphisms. A very nice paper, you should absolutely read it if whatever I'm speaking today interests you. And this is the species, the last species, Papiliopolytus, is what we are going to focus on today. This is swallowtail butterfly. Polymorphic mimicry is really, really common in these butterflies. In fact, this is one of the densest clusters of mimetic butterflies in all butterflies, especially when you consider Batesian mimicry, where one palatable species mimics a protected species. So this is Papiliopolytus. Can you try to change this at some point so that the pointer works? So on top you have the male of Papiliopolytus. Next to it in the left corner you have form Cyrus, which is a male-like non-mimetic form. And then that wing pattern, a black butterfly with a white band, has turned into these three forms, form Polytus, Theseus, and Romulus. And these three forms closely mimic three toxic butterflies in the genus Pachyliopta. So there is a very clear model mimic relationship between these species. Incredibly, all these forms occur in the same species. In fact, Bangalore, where I sit, the first, second, and fourth form occur right outside my window. I can catch these butterflies on my campus, but of course we have caught it from all across its range. I will get to that in a little bit. So what we know about this is that in a lot of these species, especially Papilio, mimicry is often female limited, meaning that males are non-mimetic, females are mimetic, and they might come in multiple forms, as seen here in Papiliopolytus. from some really nice behavioral experiments performed by a Japanese scientist, we also know that Papiliopolitus is edible to bird predators, that Pachyliopta butterflies are toxic, and then birds learn to avoid Pachyliopta arislochi if they are exposed to them at some point. And once they learn to avoid this pattern, they also avoid the mimic. They cannot distinguish between the model and mimic. So mimicry is pretty effective. What we also know is that in Betzian mimics, theoretically, is that in Betzian mimics, these mutations that confer any phenotypic similarity to these well-protected species, any mutation that takes you from that white banded butterfly to any of these is going to have a great fitness advantage. So if that is the case, any mutation that occurs in this resemblance is going to be under strong selection because of this high fitness advantage. This adaptive mutation will then spread rapidly if it is dominant, as Horden predicted. In fact, Fisher also thought a lot about butterflies. In fact, in his book on evolutionary genetics, there is one chapter just on mimicry. Everything else is, this big theme in evolutionary biology, and then there is one chapter on mimicry. So that is how important this example of natural selection has been, and a lot has been written, again, mostly in theoretical terms or speculative terms about this species as well. So there are two critical predictions that follow from this. One is that mimetic forms that evolve in succession are sequentially dominant, and the second one is the mimicry gene will show signatures of selective sweeps or other kinds of episodic selection, because every time a new highly beneficial mutation comes in, it's going to sweep through the population, right? So we decided to test those ideas. They have never really been tested, certainly not in natural populations, which is even great. So the main questions that we ask are, what is the evolutionary history of polymorphisms in Papiliopolitus? What is the genetic basis of mimetic polymorphism? I'm sorry, organizers, this screen used to be on where I could see what slide I'm looking at. All right, what is the genetic basis of mimetic polymorphism? What are the dominance relationships among these multiple forms? And has the mimetic polymorphism evolved following Harold and Steve? The first things are steps towards testing some of these ideas. All right, so before we go on, I just want to very quickly tell you that in this specific case, fortunately, we do know the genetic basis of this particular mimetic polymorphism. This came out of my early work, and double sex is the gene that controls this female limited polymorphism. We had studied this pretty well with respect to different isoforms and how they're expressed. So what is important to know is that that is the gene. Also that these mimetic alleles of Papiliopolitus are locked up in a protective inversion. So the mimetic copy is inverted, the non-mimetic copy, the allele, is in the expected direction as per the evolutionary history of this allele. So because of that inversion, there is no crossover between the mimetic and non-mimetic form, usually. Yeah? And then DSX has sex-specific transcripts that are differentially expressed in different tissues at different developmental times. So you can get the same, you can get two different forms from the same genotype, depending on where the gene is expressed. And again, remember that males never show mimicry in this species, right? So the same genotype may be in both males and females, but in males it is suppressed, in females it is overexpressed, which is how you get the mimetic butterflies. So that's just a basic which will come in handy later on. So I'm not going to dive too much into materials and methods because it's a lot of really boring and very complicated bioinformatic work. We don't have to worry about that. If you have any questions, I do have more details that I can share, for example, during Q&A. But we also did extensive breeding experiments, and the importance of that will come in handy soon. We sequenced about 82 specimens from across 6 species in this particular group. And this is the range of these species. again don't need to bother with the details. What is important to note there is that mimics overlap completely with the models. They do not occur outside the ranges of models, the toxic species that they mimic, and that every single species occurs in allopatry. There are no hybridization, there is no hybridization, no hybrid zones, nothing in these populations. They occur on distinct mainland island clusters. All right. This is again details, don't worry about that. I've already mentioned the distribution, and this is some work that Atulya did. So we use time-calibrated phylogenies to estimate times of divergence for all these species. Again, details you don't need to remember of all of this. You just need to know two particular nodes. This one here with the first blue arrow, and then this one here. That arrow is when this entire species group evolved. What you see on top is Papilioprotenor. It's the basal species, a non-mimetic species in this particular group, and everything else is mimetic here. And then you have the models in which the smallest split here is between these two. This is the model of one particular female form. This is the model of the other one, and the divergence time there is only about 0.45. So this female form number 4, which mimics Pachylepta hector, this second arrow must have evolved less than 0.45 million years. There is no other model for it. Yeah? So these are the two important time points that we identified. And what you see then is that starting from the right direction of the sequence of DSX. So the blue and gray blue is the ancestral type, and then the orange one is the mimetic type. So that inversion evolved somewhere around that time scale, and then the second form, the second type of that inversion has evolved only in Papiliopolitus. So we were able to look at the evolutionary history of this species only because of the peculiar structure in which all these forms are distributed. I will get to that separately. Yeah? So, as predicted by Haldane, what we did find, and this is after a lot of breeding experiments, is that the most ancestral form is the non-mimetic one that evolved somewhere close to 9.6 million years ago, and then the first mimetic form evolved along this branch going from about 9.6 to 4.2 million years. We don't know exactly when it evolved there. There are ways to look at it. We haven't done that yet. But that is the time window in which that evolved, and then form Politus is the latest mimetic form which evolved about 0.4 million years ago along that branch. And if you look at the segregating broods, segregating back crosses that we perform, and I'm just showing this as a casual one line, but this is more than 3 years of work by Riddhi and I. Anyway, the blue arrows here show genetic dominance. So the oldest form is the universally recessive one. The youngest form, the mimetic one, is the universally dominant one. And this form is intermediate between those two in terms of dominance. So this was a very nice finding. How much time we have? 2 minutes. Okay. And then 5 minutes of Q&A? No. Okay. All right. And then you see sequence variation. Don't worry about it. What is important to note is that the 3 alleles of the 3 forms are very, very well distinguished in this. We have markers which also look at those. And what is pretty remarkable in this is that you have these 3 DSX alleles which are marked by really rapid molecular evolution. What you see here is the split from the basal Papilioprotenor. Here is the first mimetic form and second mimetic form, the youngest stages of these. And what you see here is nucleotides and amino acids. There is absolutely no change in the basal form, the non-mimetic form. But the moment you get the mimetic forms, there is extremely rapid evolution of both nucleotide sequences as well as amino acids. We do not believe that all of these are under selection. Some of these may have just spread in the population. They might have hitchhiked, but what you do see is that there's tremendous rate of divergence between all these, and even amino acids have been changed even in the domain regions, which is pretty serious. This is a gene which is important in sex differentiation. So it's a highly conserved gene. We have looked at the evolution of this gene over 350 million years. You do not see much change in domain regions, but even among these female forms, some of those changes are in the domain region. So this is a big hotspot for molecular evolution. And in fact, if you look at those, this is Papiliopolitus, 3 forms in a single species, and this is the genera and families and genera across which we are comparing variation in this. This is really, really very high. The variation you see among 3 forms is more than what you see in most genera of entire insects, including some big ones like Drosophila and so on. So this is crazy in terms of evolution. This is 115 substitutions, about 13% of the entire exonic region. So what you see is that, as expected, you have tremendous signature of episodic positive selection on this particular gene. Each ring there is a different species, the 5 species in this mimicry ring. Every single one of them, every single one of the species had selective sweeps in DSX. Yeah. So that is pretty interesting, and the number of loci which are within DSX which are showing selective sweeps are also pretty high, but they're not related to the range size and clade age. So this is happening independently of that. All right. The next one is regarding developmental genetics, so I'll skip that and I will go directly to conclusions. So the first thing is that female-limited mimetic polymorphism is old and under complex selection regimes, as seen from the allelic histories. It does follow Halden's sieve of this successive dominant forms. It has experienced multiple selective sweeps in all species, but not the basal non-mimetic species in the clade. Many of the genes having experienced selective sweeps outside of double sex also contribute to wing development, color patterns, and related traits involved in polymorphic mimicry. So it makes sense why these species have experienced that, and all not substitutions are probably involved. Some of them might have just hitchhiked. That is a little bit complicated. If you are interested in knowing about this, I will come to that later. All right, what we have not shown is a longer list, so I'll skip that for now. But we are really looking towards looking at selection and regulatory regions, do some selection experiments in the field, look at allele frequencies and selection coefficients in natural populations, look at life history experiments. Some of this will likely publish next year. But if you're a student with ambitions in evolutionary biology, bioinformatics, and big genomic data sets, talk to me. We have a lot of it. We just need very good students to analyze all of that. Again, big thanks to my students, a bunch of people who have helped with sample collection and sequencing. Thanks a lot for all of that. And I will just point out that if you want to read more about this, my lab is biodiversitylab.org. Our Butterflies of India website is very popular. That's ifoundbutterflies.org. We have a very nice butterfly brochure which covers all the species here if you like, or any city that you are in, this covers all of those. We have extensive collection which allows us to do this kind of work all the way from taxonomy and systematics to genomics and molecular evolution because of our collection. So you can read more about that at Biodiversity Collections. And IBMS network is for Butterfly Monitoring India. That's something that we have been spearheading for the last more than 16, 17 years. So more about that maybe during the break. Thanks a lot.
So any question from the audience?
Yes.
Different ecological conditions the intensity of selection would differ. But have you seen any mutation rate cases? Difference in mutation rates.
In different selection regimes. The question is, have you seen mutation rates? Have you seen changes in mutation rates based on ecological conditions? So there is a distinction between mutation rates and substitution rates. Mutation rates we can't estimate, substitution rates we can. This is substitution, by the way, for stewards who do not know this, is what gets in the population. Mutations might come and go, substitutions remain out of, so it's a subset of mutations. So that we can estimate, we haven't done it under different ecological conditions. We in some sense try to address that by looking at substitution rates and loci under selection in DSX and other places, the last table that I showed. But there it doesn't show any pattern as to whether it's related to population size, whether it's related to range size, or island, mainland, different kind of habitat types and so on. But it is logical that there would be some relationship, just not in this particular group. Yes.
The dominant mutation, you said it goes to a sweep.
Mm-hmm.
How do you expect a polymorphism now?
So there is the reason why we are able to do it in these polymorphisms is because the polymorphism itself is under selection. So the reason why in other cases you do not see wild type is because it gets swept out. Yeah? It just replaced by the new mutations. Here the non-mimetic form has some adaptive significance, which is why it is not completely taken out of the population. So the new mutation rises in frequency, but then its rise is limited at some point because of this balancing selection. And there have been ideas about whether it's because of sexual selection or some kind of natural selection, physiological differences between different female forms and so on. We have done tremendous amount of work on that in the last 15 years. This has been a staple of what my lab does, and that is the paper I mentioned we might publish next year. But we have looked at that tremendously, and we
At a
understand.
certain point, there will be an
Sorry.
equilibrium, is it?
Hmm?
At a certain point, both these forms will be in equilibrium.
They will be at some equilibrium. It doesn't have to be a stable equilibrium. It can be a dynamic equilibrium, but that is what it is. And the data we have shows that that equilibrium point is decided by how common the model is. So if the model is rare, the form is rare. If and then the mimetic form cannot completely take over the population. If the model is really common and the mimic is relatively rare, that negative frequency dependent selection.
A small questions.
If that is the case, then the mimetic form completely takes over. But in most of India, that is not the case. There is some balance.
You were speaking about the double sex.
Yeah.
There are some changes, significant changes in some domain.
Right.
Will it affect the sex ratio?
Sex ratio itself will not be affected by double sex. Yeah. Not unless there is some selection on it. So double sex has two kinds of isoforms, male and female isoform, which are again regulated in some, and you don't have to get into the details of that. That's actually given really well in Saurabh Baral's paper. So double sex itself will not change the sex ratio, and there are some species in which sex ratios are known to be highly biased towards one sex or another. Again, double sex doesn't have anything to do with that. So we do know that double sex is capable of making male and female bodies, but not of changing sex ratios under a certain allelic composition of an individual. Yes.
Mentioned about inversion.
I mentioned who?
Inversion.
Huh.
Inversion, we have written that inversion has occurred. Inversion.
Inversion. Okay. Sorry.
Which type? There are two types of inversion,
Right.
paracentric and pericentric. So
Right.
you are referring to which type?
So I will tell you what it is. It's a complete inversion, yeah. And this inversion is really interesting because it narrowly borders the double sex gene. So it just locks up that particular gene and nothing else outside of that, yeah. And what that does then is that, as I mentioned earlier, the mimetic and non-mimetic female forms do not cross over because of that. What is interesting is that both the mimetic forms, and I didn't show you that data set right now, both the mimetic forms have the same inversion. Yeah? So they could potentially cross over. And still the crossing over in these two forms is very, very rare. What is causing this cold hot spot or cold spot of
You know that inversion suppress crossing over. Inversion suppress crossing over.
so that is what I'm telling you. So inverted copies should still be able to cross over, and they still don't. Whether there are some smaller inversions within that larger inversion is not something that, they're certainly not in exonic regions. There may be in intronic regions, and intronic regions in this particular gene are very long sometimes, 10, 20,000 bases in the longest intron. So that is something we do not know, but that's the most interesting thing about the inversions, that you do not have this. We did find a novel form, and that's again something you can see in the paper that just came out actually last week, so you're able to see the details of all of this, is that there is one crossover in exon 1 and exon 2 to 5, which produces a completely different form, which takes forewing of one form and hindwing of the other, and that is again very rare. This is the first demonstration that just one exon controls one wing and other exons control other wings, again following the dominance hierarchy, but you just need first exon of the dominant form to change the forewing. That's
Okay.
also pretty
Okay.
interesting.
Thank you.
Thank you.